this week. Okay, thanks, Sarah. So it's an absolute pleasure to introduce Rebecca Dewhurst-Trigg, less formally known as Bex in the Islet Biology Group and at Exeter. So Beck joined the IBEX team in 2021, so just over 3 years ago, after completing her PhD looking at the influence of dietary fats on adipose inflammation. And she joined the groups of both myself and Martin Eichmann, where she contributed a huge amount of work to a number of different research projects, but today is going to focus on a project that's currently funded by JDRF. It's 1 year into a 3-year project grant awarded to myself, Sarah and Noel, and it is looking at the islet protective role of pancreas MSCs in type 1 diabetes and in health. So yeah, when you're ready, Beck, take it away.
Thanks, Chloe, for the introduction. Hopefully everyone can see my screen there. I'll just double check before I start.
Yeah, that's perfect, thanks.
Thanks, Ellen. Great, so I'll get started. So mesenchymal stem or stromal cells, or MSCs as I'll refer to them throughout this talk, exist in virtually all vascularized tissue, including in the pancreas. Not one single marker can identify or define an MSC, but they are positive for markers such as CD90, CD105, and CD73, and they lack chemopoietic markers and CD31. The therapeutic potential of MSCs has been widely studied in culture-expanded exogenous MSCs, and MSCs have been shown to have both immunomodulatory and regenerative functions. And it is the secretome of MSCs that is believed to be the main mechanism through which they achieve their therapeutic effects. Indeed, in clinical trials, beta cell function has also been preserved by infusions of MSCs in patients with newly diagnosed type 1 diabetes. And they have been shown to home to inflamed tissues and have the capacity to migrate towards pancreatic islets themselves. However, we know little about the role of native endogenous human pancreas MSCs and their role in both health and in type 1 diabetes. Therefore, the aims of our studies was to determine whether the number, distribution, and islet protective phenotype of native human pancreas MSCs is altered in type 1 diabetes. And we will look at this in situ. And in vitro, we would aim to determine whether MSCs can withstand the cytokine stresses seen during type 1 diabetes pathogenesis. And we hope to identify novel islet protective factors in MSCs that have been co-cultured with cytokine-exposed beta cells. And these studies collectively will hopefully provide us with scientific information on MSC-related changes in type 1 diabetes that we may be able to target therapeutically in the future to help delay or prevent the progression of diabetes. Moving on to our methodology, we had human pancreas tissue, so formalin-fixed, paraffin-embedded human pancreas tissue from the Exeter Archival Diabetes Biobank, EADB. And we immunostained these sections, and I'll go through the methodology of this now. So we stained our sections with opal technology, which allowed us to scan whole slide scans on our phenomimager. We were able to stain 6 markers on each section, and we had 2 serial sections, one with our MSC panel, which I will go on to describe in the next slide, and also our islet hormone and immunomodulatory factors on a serial section. We were then able to scan these serial sections and fuse them together in our software called Halo. We were able to identify our insulin-containing and insulin-deficient islets. We then were able to determine the areas of tissue on the serial sections that overlaid, which we could then take forward for our analyses, as we'd have both our MSC and our hormone panel on this tissue area. We were able to identify our cell phenotype of interest, our MSCs, for example. And then we were able to perform spatial analyses looking at the position of our MSCs in relation to islets or to other cell types such as beta cells, for example. So if I just go on to our MSC panel, we used 6 markers to identify our MSCs in situ. Our phenotype of MSCs was cells that were positive for CD90, CD105, and CD73, and negative for CD31, CD34, and CD45. We also had a core panel initially that consisted of just 3 of these markers, CD90, CD105, and negative for CD31. So we wanted to have a look at whether it would be possible to identify our MSCs accurately with 3 markers, which may then leave other areas of a 6-plex panel for optimization of other factors. And we could see that both the 3 and the 6 markers correlated extremely highly, as you would expect, considering our 3 markers were also included in our 6-marker panel. However, as you'll notice from this correlation, the total number of cells was quite different, showing much less cell number for a 6-plex panel, which just shows the importance of having all 6 markers in our panel. Therefore, we carried this forward for all of our analyses, including our 3 positive and 3 negative markers to identify MSCs. And this is what it looks like in the tissue. So we have our 3 positive markers here, CD90, CD105, and CD73, with an overlay on the right-hand side. And these white arrows in the micrographs show our MSCs. And here is our 3 negative markers, CD31, CD34, and CD45. And this is our insulin and glucagon staining from a serial section, showing this is an insulin-containing islet. So following our analysis, we looked at the number of native pancreas MSCs at the islet periphery. And we saw there tended to be an increased number of MSCs around the insulin-containing islets of individuals with type 1 diabetes compared to those without diabetes. And in the individuals with diabetes, we saw a tendency for an increased number of MSCs around insulin-containing islets compared to insulin-deficient islets. And because of our ability to look at whole slide scans, we were able to look at a large number of islets per donor, here in the table showing 100 to 150 islets per donor. And these data are part of our ongoing studies, where we aim to investigate the number of MSCs, the distribution of these MSCs, and their islet-protective phenotype. And we will add an additional 21 donors to this data set from the NPOD biobank. So that's ongoing studies that we're currently performing. We also know that MSCs secrete annexin A1, and it's highly expressed in human MSCs. And exogenous annexin A1 can potentiate glucose-stimulated insulin secretion and protect from cytokine-induced cell death. So we wanted to know whether our MSCs at the islet periphery in situ were also expressing annexin A1. So we were able to look at this. Again, going back to our MSC panel, our positive markers, CD90, CD105, and CD73. And our negative markers for MSCs, CD31, CD34, and CD45. So they were on one section, and then on a serial section to this, we had insulin and glucagon to identify our islets, and annexin A1. And as you can see, we can see annexin A1 staining around the islet, and the white arrows here indicate the annexin A1 positive MSCs that we see. And again, we can see these at the islet periphery. Looking at the number of annexin A1 positive MSCs, again, we saw a tendency for an increased number of annexin A1 positive MSCs in individuals with diabetes compared to those without. And in individuals with diabetes, we saw that there tended to be an increased number of annexin A1 positive MSCs around insulin-containing islets, or ICIs, compared to insulin-deficient islets, or IDIs. However, the percentage of native human pancreas MSCs that we saw that were expressing annexin A1 was similar in individuals with and without diabetes. And this is consistent with our in vitro data, where we exposed our MSCs to cytokine conditions that were reflective of type 1 diabetes pathogenesis or islet transplantation, and our no cytokine control, where we saw a high expression of annexin A1 across all of these groups. And again, these data are from ongoing studies where we aim to add an additional 21 donors with and without diabetes from the NPOD Biobank, which is currently being performed. So for our in vitro experiments, we had culture expanded MSCs, and we exposed them to cytokines reflective of the early or later stages of type 1 diabetes pathogenesis or islet transplantation. And we wanted to look at the viability of these cells under these conditions. So we exposed our cells to these cytokines for 24, 48, and 72 hours. And here's a graph showing the percentage of live cells or the percentage of live MSCs. And here's where our no cytokine control group sat. And as you can see, the viability of our MSCs is similar across groups. And if we take those groups that show our early and later stages of type 1 diabetes pathogenesis, we can see that MSCs can withstand the cytokine exposure reflective of these conditions. In addition, we know that MSCs can respond to specific cues within their microenvironment, and they can secrete cytoprotective and immunomodulatory factors. We also know that MSCs can improve islet function, and this can be enhanced in particular by direct cell-to-cell contact. However, we know little about the mechanisms through which MSCs and their secretome influence beta cell inflammation, and this is something we're also looking at with our in vitro experiments. We are looking to identify novel islet-protective factors in MSCs that have been co-cultured with cytokine-exposed beta cells. So I'll go through the methodology for this experiment here, where we have our MSCs and our endo-C BTH1 cells, or endos, which is a human beta cell line. So we want to culture these in direct contact co-cultures and then separate out our MSCs so we can perform RNA sequencing on these cells. So initially, we labeled our cell types with 2 different dyes, so we were able to distinguish these cells apart. And we plated these together in direct contact co-cultures. We then used FACS to separate out our cells so we could take our MSCs for RNA sequencing. So we identified our MSCs, which were labeled red, and our endos labeled yellow. For our MSCs, we were able to get on our cell morphology and then collect our live cells. For our endos, we found our single cells and again collected our live cells. Looking at this direct contact co-culture of our endos and our MSCs, if we look at just the endos alone to start with, the graph on the right-hand side, we can see that following cytokine exposure for 24 hours, our beta cell viability was reduced, which is what we hypothesized and what has been previously published in the literature. However, when we co-cultured these beta cells with our MSCs, this reduction in viability was attenuated following co-culture. So our MSCs were able to improve beta cell viability. So these MSCs that we have seen to improve beta cell viability were collected by FACS, and we are currently in the process of our RNA sequencing to identify novel islet-protective factors from these MSCs that we can hopefully go on to use in both in vitro experiments to understand how they may be helping beta cell viability, but also to look at these factors in situ in a human pancreas and whether the MSCs in the pancreas also express these islet-protective factors. In summary, our in vitro experiments, MSCs can withstand cytokine exposure reflective of the early and later stages of type 1 diabetes pathogenesis. And MSCs can attenuate the cytokine-induced beta cell death in direct contact co-cultures. And we have our ongoing experiment with these MSCs to perform RNA sequencing, which we aim to identify novel islet-protective factors that we can then hopefully look at in situ to see whether our MSCs in situ are also expressing these factors. For our in situ data, we are able to identify native human pancreas MSCs with our 6-plex panel in both individuals with and without diabetes. And in individuals with diabetes, we see a tendency for a reduced number of MSCs around insulin-deficient islets, but retention around insulin-containing islets. We also see that these MSCs at the islet periphery express annexin A1, which is islet-protective. And this data is part of ongoing studies where we aim to add an additional 21 donors with and without diabetes from the NPOD biobank to our current data from the EADB biobank. So in summary, our native human pancreas MSCs can be identified in situ, and they appear to form a border around insulin-containing islets. And we see that these MSCs express annexin A1, suggesting an islet-protective role. I'd like to thank everyone involved in these studies, particularly those in the Rackham group and also the Islet Biology at Exeter group for all of their help and support for these studies. The JDRF funding our in situ experiments, and Diabetes UK for our in vitro experiments. And of course, all of the donors and their families from the EADB and NPOD biobank, without whom we could not conduct this research. And thanks everyone for listening.
Brilliant. Well done, Beck. Really nice, clear presentation. So lots of rounds of applause I can see up there. So let's open it up for questions for Beck. I'm trying to make sure I can see all the hands if they're coming up. Please put questions in the chat if you want to as well. Okay.
Can I ask a few?
Yes.
I don't know anything about MSC type questions. Say that difference between the 6-plex panel and the 3-plex panel is quite striking. Do you know what the cells that are excluded, like between the 3 and the 6, actually are?
They could well be other stromal cells or stellate cells. They could well be parasites, which we know have a close relationship to MSCs. So I think it could be those, but
yeah. Oh, interesting. And Annex A1, do we know that much about actually what it does? I know nothing about it, so just wondering.
In the context of islet function, I think it can improve glucose-stimulated insulin secretion. So I think it's islet protective in that effect. And I think it can also improve like cytokine or reduce cytokine-induced cell death.
Okay.
I think, yeah.
Great, thank you.
No, I think you've got a question.
Yeah, thanks. Well done, Beck. That was really nice. Can I ask about the staining of annexin A1? It did look as though there was quite a lot in other acinar cells in the pancreas. Is it a product of a whole host of pancreatic cells as well as MSCs, or where does that other stuff come from?
So I think annexin A1 can also be expressed by fibroblasts or ductal cells. It can also be expressed by T cells and endothelial cells. So I think it could be a combination of those. And it may be possible for us to identify other cell types with our 6-plex panel that do also express annexin A1. So it could be something we could look at further.
Mm-hmm. And have you noticed, maybe you haven't had a chance to do enough analysis yet, have you noticed whether there's any difference in that, what you might call the remaining pancreatic annexin A1 in controls versus people with type 1?
It's not something I've noticed yet, but it's something we could definitely look at in the future. I know annexin A1 itself can be different in the plasma of patients with diabetes compared to those without. So it's definitely something worth exploring in the future, I think.
Good. Thanks, Beck. Yeah, well done.
Yeah, I guess at the moment you're doing a yes, no, have they got an X in A1? I think as you develop the pipeline a bit more, you might actually be able to look at levels, which, will expression levels, which I think will be interesting.
Yeah, definitely.
Yeah, perhaps I can just add one other question. So you mentioned in one of your earlier answers about the different types of cells that could be there, the pancreatic stellate cells. Now, they seem to be quite an interesting population. Do you think you can define those with your panel?
That's a good question. I think I need to understand the markers that they're positive and negative for in more detail in order to know. But I think having our 6-plex panel and also our pipelines to have our serial sections overlaid may give us the best chance of identifying other cell types that do require multiple markers to identify them. But yeah, I think it's something I need to look at. I think a lot of sort of stellate cells and like fibroblasts, for example, even parasites, I think a lot of the markers overlap or different cell like phenotypes within that group of cells can express or not express the same markers. So I think it's actually a really difficult apart. But hopefully, again, as you mentioned, looking at the expression levels may well give us an insight into these different cell types.
Yeah. And I think another area that will be of interest, and perhaps is something that we can help with as well, is as there are more spatial transcriptomic data sets that come out, we might be able to start looking a little bit at what's the MSCs are expressing in the vicinity of the islets as well, and see if we can translate that through into protein using your beautiful staining, which I think would be great, and perhaps then pick out some of those different supportive or not supportive cell types, depending on which way we look at it, that may be around the type 1 islets.
Yeah, I think so. And hopefully our RNA sequencing data will really help understand how those MSCs of the islet periphery may well be supporting the beta cells.
Yep.
So being able to translate that into protein would be really interesting, I think.
Yeah, excellent. That sounds good. Have we got any other questions that people would like to ask of Beck? Just give everyone a few moments to consider. If not, thank you very much, Beck, for an excellent presentation. We really enjoyed that. Good luck for your sugar science one in a few weeks, isn't it?
Thanks. I think probably more towards the end of autumn, hopefully.
Okay.
When we've hopefully got a little bit more data, so we can share a little bit more of the story.
Excellent. We'll try and share that when that's coming out as well. And I'm going to hand back to Anna, if that's all right, just to give us reminders as to which when our next T1D egg session is, and just to encourage people, if you would like to speak at one of these sessions, please do reach out to Anna and I, and we can try to schedule a session in the upcoming plans for these. Oh, and Noel's just put a great point in the chat about Jo Balderson, who is on Sugar Science tomorrow. So if you have an opportunity to tune in and support Jo, I think that would be great for her to see lots of friendly faces. So thanks, Noel.
Oh, I thought I'd get away with it.
Sorry, Jo.
Oh, well.
You'll be great. We're looking forward to it.
I'm furiously putting together slides.
Well, good luck.
Thank you.
Well, that's exciting. I didn't know about that, so now I can tune in. Yes, so our next T1D egg is on Monday, the 8th of July, 1 to 2 p.m. again, so same time slot. And then we're going to have a couple of months of break in August and September, and I think we'll be coming back in October. So we'll send out some details for that nearer the time. Yeah, I think that's everything. Thank you so much, Bex. That was a great talk, and yeah, and thank you everybody for coming along.